Follow us on social media and please hit the subscribe button so you don't miss out on any updates from our ALS experts.
Hello, folks. I'm McFinn Levere from Everything ALS. It is so good to see your faces and your names. And so grateful for the inventor of Zoom to bring us together. There's a lot of things to be grateful in our life. And tonight, I think we should be grateful for the researcher that comes onto our program and gives us those little tips that took so long to gather. And I'm not sure how long we're all going to be on this journey together, but I am so grateful that each one of you come every other Wednesday, participate, and bring your little treasures to share, because we might have little gifts that we don't understand how much of a gift they are to people who haven't figured out that problem yet. So please, tips and tricks, they're always valuable. And when we have our open forum at the end of this talk tonight, please, if you have anything, sing out with it. And if you have any needs, sing out with those too, because we're here to help. All of us, each one of you. Team, thank you so much. And Lisa, it's all yours.
Thanks, McFinn. Welcome, everyone. It's always great to see lots of familiar faces and new faces as well. I am Lisa Deegan, and I'm part of the Everything ALS team. And we love to be able to provide the ALS community access to the top ALS experts around the globe. And we also love to connect the experts directly with our community to address their questions. So thanks again for being here. And after the presentation is done in the Q&A portion, this is where we open up, turn off the cameras. It's a safe place, like McFinn talked about, talking about whatever's on your mind. It's a way to connect with people going through a similar journey. So thanks. I'm glad you guys are all here tonight. So for anybody who is new, just really briefly, I'm going to just share, because sometimes people say, well, what does Everything ALS do? I don't really understand. So I'm just going to give a brief description of what we're about. So we are a nonprofit, and it's built by people all affected by ALS. And our focus is advocacy and research. So we're kind of at the cross section of where the patient, the caregiver, medical community, the FDA, pharmaceutical community, and we're conducting citizen-driven research that I had mentioned earlier, and open data innovation, and we're hoping to change the paradigm in ALS. And open just means we share. So we're not working in a silo. So we do leverage technology for digital biomarker discovery to be used as a measurement tool for early diagnosis and progression tracking and advanced therapeutics. So we have created this open scalable platform that allows collaboration. Collaboration is key because nobody can do it alone. So again, I had mentioned our research is really aiming to detect ALS earlier. The earlier you get it detected, the earlier you're on treatments, and better for patient outcomes. So if you want to get involved, you can send us, or have any questions, just send us an email, info@everythingals.org, and I put in the chat how you can get involved in our research. So let's get to the exciting stuff. So tonight we are so honored to have Dr. Michael Snyder, who's going to be presenting on his latest research on discovering an architecture of genes that are potentially linked to ALS, and the insight that this research provides about the disease. So Dr. Michael Snyder is a professor in genetics at Stanford University School of Medicine. He's a world-renowned leader in the field of functional genomics and proteomics, and one of the major participants of the ENCODE project. So earlier this year, Dr. Snyder authored a paper with his co-leads, Dr. Zhang and Dr. Cooper-Knock. Thank you for joining us as well, Dr. Cooper-Knock. And it was published in January on I think the 18th in Neuron. So this research opened up a whole potential of new genes to explore. So I'll let him talk to you more about that in depth, but we're super excited. I'm excited to hear what he has to say. So please join me in welcoming Dr. Snyder.
Great. Well, thanks very much for having me here. I am very new to the field, so I'll probably learn more from you than you will from me. I am going to tell you about our recent work on the genetic architecture of ALS. it will be pretty science geeky. So feel free to jump in and stop me and interrupt if I get caught in jargon or say something that you don't understand. So the outline of this talk is I'll give a super brief background on ALS and the genetics and in general. I want to tell you how we got into this field. It stems from our work doing big data research in general. And then I'll tell you about the results we got from the study that was just published. So most of you probably know this better than me. Certainly Jonathan Cooper, my collaborator, I forgot to say at the outset, two key people involved in this project are Sai Zhang, postdoctoral fellow, now instructor at Stanford, and Jonathan Cooper Knock at the University of Sheffield, and then a whole team of others who helped us with this, including the members of the consortia that have contributed samples and data, et cetera. So, okay, so here we go. ALS, I'm sure you all know, it's a neurodegenerative disease that weakens muscles, and it's thought to mostly involve motor neurons, which die and lead to death from respiratory failure. The cause of this is unknown, and there's no cure. In spite of this being quite common, 1 in 300 people have a lifetime risk of this. And the fact that it's very severe, 20 to 48 months of survival after diagnosis, it's pretty amazing that we know so little about this disease. And so I think we'd all agree there just needs to be a lot more effort to try and solve this for all the reasons you heard earlier from earlier statements. So I think you probably all know that most diseases are a combination of genetics as well as environmental factors and lifestyle, which is often lumped into the environment. So your genome certainly can lead to high risk for disease, but there are various environmental exposures, chemical and other pathogen exposures can lead to this. I don't know if COVID actually leads to an increased stress. Food, exercise can all either accentuate or mitigate disease onset. And so people have tried to quantify this, and it's highly controversial how much of a disease is caused by your genome versus these other things. And it probably depends a lot on the particular disease. Some people say overall 25% of your disease is contributed from your DNA sequence, but and the rest comes from other parameters. But that will be, as I say, highly gene dependent. In the case of ALS, they've measured this, and you can measure this using twins. So identical twins have identical DNA, as you probably know. And then fraternal twins, they share DNA, but it's not identical. They share half their DNA. And so you can do genetic statistics and look at incidence of ALS and make calculations as to how much of the disease is due to genetics and how much then comes from other things, meaning environment and lifestyle. And when you do that from the study here, the number is about 61%, meaning there's a very, very strong genetic component to ALS. So it's caused to a large extent by genetics, although other things clearly contribute as well. In spite of that, if you actually look at the genetics of ALS patients through sequencing or other means, typically less than 10%, you can find the underlying gene. So that means we haven't solved most of the gene structure. We don't know the genetic basis of this disease. We know there is a genetic component, but we don't know the genes involved. And that's really the background for our study. You might say, well, why do you want to know the genetics of a disease? What good does that tell you? Well, often when you actually reveal the genetics of a disease, you can find often in complicated diseases like this, hundreds of genes involved, and they tell you something about the biology of the gene. And just to give you a few examples of some unexpected findings, this comes from Manolis Kellis's lab at MIT. They were looking at Alzheimer's, and when they actually looked at the genetics of Alzheimer's, they discovered it had a very strong, a lot of the genes were involved in immune function. That was a big surprise because I think people are thinking Alzheimer's is a brain disease, but it turns out there's a strong immune component. Now it's very clear Alzheimer's, it is an immunological disease, at least at some level. We did some genetic architecture work on autism a bit ago, and actually everybody's looking at a certain region of the brain. It's deep cortical neurons. And that was found, but we also found a whole new region of the brain that was involved that made a lot of sense as people dug into the disease called the corpus callosum. So by doing the genetics, we can learn something about the disease. We can also learn things about mechanism of the disease, what might be underlying causes and things. If you get at the genetics is generally thought to be upstream of the disease and can give you clues as the cause. And a good example of this, many of you may know ALS obviously has a lot of protein aggregation as one of the characteristics of ALS. You see these aggregates of protein, and I'll talk about 1-TDP-43 a little bit later. And so a lot of people say, well, these aggregates, they're really causing the disease. Other people say, well, no, maybe they're just gray hair. Like gray hair doesn't cause aging. Gray hair is a product of aging. Nobody thinks that you get gray hair and that causes you to get old. It's the other way around. So by getting into genetics, you can try and get underlying causes, at least upstream events in the disease. And that's another good reason for trying to decipher the genetics. And lastly, if we get good at this, this hits nicely with I think the mission you guys described about ALS getting early markers. Well, I would argue one of the earliest markers is your genome. If you can predict from a genome sequence who will get the disease, that's very, very powerful, obviously, into taking early interventions. And that is proven over and over again. If you can intervene early, you have a much better chance of mitigating the disease rather than intervening late. So for all these reasons, it's very useful to underline it's useful to decipher the underlying genetics of complicated diseases like this. So our lab is known for actually doing big data, and we've invented a lot of technologies that are out there for trying to decipher complex biological problems. And so we do things like, we didn't invent genome sequencing, but we actually are the first to apply it for looking at healthy individuals. We also use technologies for profiling literally hundreds of molecules in your blood and urine, or sorry, thousands of molecules, I should say. And more recently, and I'll just touch on this in the end if there's time, but we think wearables are extremely, extremely powerful for for onset of disease and disease progression. I'll show you some examples of that later. And so one way we think about this is if you look at the way medicine's done today, if you go to a doctor's office, they usually measure anywhere between, say, 10 and 20 things. And some of those things are actually not very useful. And yet you're trying to get a profile of someone's health or disease. 10 or 20 things, we would argue, probably doesn't cut it, especially in an era where you can get literally, tens of thousands, if not millions of data points on people. And this is what we do. We literally take billions of data points on people. You'll see in a minute. And so I like to use this analogy here. If you're trying to get a profile of someone's health, and you have imagine you have a jigsaw puzzle of 1,500 pieces, getting 10 or 15 pieces isn't going to give you a very good picture of that puzzle. But if you can get 1,000 pieces in a 1,500 puzzle, you'll get a much better idea what that puzzle looks like. And that's how we view the health profiling we do. We're trying to get as many pieces of that puzzle to get as complete a picture of your health as possible. And we do this in a fashion that lets us detect disease early. And so a major flagship project in our lab is to do deep profiling on people. Some of you may know this, maybe not. But we actually sequence people's DNA, and then we make as many molecular measurements out of their blood and out of their urine. So we'll follow all their RNA and proteins and metabolites, all these molecules that are in people's blood cells, and actually what's called the plasma, the part that doesn't have the cells. And that gives us a window, if you will, into the molecular profile, what's going on in a person. We also, believe it or not, study their poop, their microbiome, in their gut, and we do deep clinical testing and lots of advanced tests as well. And we also are very, very keen on wearables, as I'll talk about that in a minute. And so we've been profiling a set of people. Well, I'm actually the first guinea pig. We've been profiling me for 12 years, and 109 people we've been following for now 9 years. So we sample them every 3 months when they're healthy. And if a disease comes along, we'll take many more samples to see what's going on. And we're doing this to try and understand what it means to be healthy, how does it change over time, and can use these advanced technologies to get a window into people's health, if you will. And so with that in mind, it turns out that just to give you, again, giving you a lot of background, but you'll see where this is going. just from these deep profiling. So in this 109 people, they were generally healthy people. And we're collecting literally billions of data points as we follow them over time. And basically, what we discovered is that these advanced technologies like sequencing people's DNA and profiling all these molecules led to a lot of health discoveries, all before they had symptoms. It relates to one of your missions, trying to actually find disease pre-symptomatically. And it was a wide range of diseases because these are healthy people, but nearly half the people, 49 out of 109, learned something pretty important about their health. Like in the heme-oncology space, we found someone who had early lymphoma, and 2 people with these are called precancers. MGUS, they can turn into aggressive cancers. Several people with cardiovascular disease, and some from genetics and genomics, and others from actually wearables, metabolic, and so on and so forth. So no one technology found all these things. That was the power of using all the different technologies. And so we got a much more complete picture of people's health, and then again, could see early signs of disease pre-symptomatically. And in actually all these cases, we were able to intervene productively, even caught my Lyme disease of all things from a wearable. That was me. And one area that was powerful though was the genome sequencing. So we actually did sequence everybody's genome, the 109, and of the first 70 people sequenced, 12 learned something pretty important about their genome. Now, these are mutations that actually, as single gene mutations, they can cause disease at a high frequency. And ALS, there's a few examples like TDP43 you may be familiar with, and a few others, that mutations in those genes can cause ALS on their own. The classic one is here. That this, you've all probably heard of BRCA, that single knockout mutations in this BRCA gene lead to breast and ovarian cancer women, a very high frequency. Half women with these mutations will get this disease. And so knowing this is actually very powerful. And so again, a lot of people did learn something important. And in our case, we actually covered this person had a heart defect that was figured out from his genome sequence. This person had a mutation here, and they were prone to certain cancers. They did whole body MRI, and sure enough, they actually had early thyroid cancer. That was caught and cut out. So genome sequencing was quite powerful, but this is in a very special case. These are these cases where single gene mutations can lead to the disease. So it's not common. You can see it's a minority of cases, 12 out of 70, we could find their risk. There are other kinds of disease that you may have heard of. So they call these single gene cases Mendelian disease because they're very easy to follow in how they pass through their family. But most diseases that are out there, including ALS, so there are ALS examples that are single genes, but most ALS is actually what's called complex disease. And these are thought to be due to, rather than single gene mutations that cause the disease, they're thought to be changes, usually in the control sequences of genes, that by themselves cause a very, very tiny effect. And then large numbers of them actually is what's caused the disease. So the analogy is, death by 1,000 cuts here versus these single genes. You can think about just killing gene with a sledgehammer and leading to the disease. And most diseases, again, are thought to be this way. I listed here schizophrenia, but coronary artery disease, diabetes, most common diseases are thought this way. And these are much harder to figure out. So what people have done is they've done something called polygenic risk scores. You actually can sum up all these little changes. And these days, people use thousands, if not millions of these changes that they sum up to try and predict your risk for these complicated diseases. And it kind of works. It works just for the top end, the top 3% of people who have this. Most people, it doesn't really work. But this is how people operate. And there's 2 problems with this. So now, is this getting too sciencey and geeky for everyone? Are you guys able to?
No, I think this is great.
No,
Okay, great.
yeah.
Okay, and then I'll pause for questions in just a minute. But what people do is they sum up over the literally millions of these tiny little changes. And if you have all the bad ones, you're at high risk. And some people have protective ones and they get at low risk. And believe it or not, I actually predicted my type 2 diabetes this way. I'm in that top 3%. So it worked on me for diabetes. It doesn't work on most people. And there's two reasons for that. One is that genetics isn't linear. And what I mean by that is you can't just add these things up. It doesn't work that. It's more complicated. The 1 + 1 is not 2 in genetics. 1 + 1 can be 1 or it can be 15, depending whether these things, when they act synergistically, they can cause bigger problems than just the sum. That's problem number one. And problem number two is it ignores all these uncommon rare variants that are there of large effect. It's only dealing with the tiny little effect changes, if you will. And so we needed new ways to analyze genomes. And so this is where I'm leading into, I'm going to show you an example we came up with another disease, and then I'll get into ALS. But are there any questions about this so far? This is kind of the background for how we got into the genetics of this. Does that all make sense? Do people have questions about any of this?
Yeah, Dr. Snyder, we'll do all the questions at the end because people are just putting them in the chat. So if you want
I see. They're piling up.
and we'll just do them all at one time, that'll be great. Thank you.
All right, I just looked at the chat. Yep, you're piling up some good ones here. Great. All right, let me move the chat. I just blocked my view. Okay, great. Yeah, keep those questions coming. So we decided to try new ways of analyzing genomes. There are new methods you may have heard about, right? You've all heard of artificial intelligence. Well, believe it or not, you can use that for analyzing genomes. And this is what Cy did along with some other folks in the lab. They came up with new ways using these new artificial intelligence. They're very computationally intensive, but they really give insights, and they don't count on linear effects as this just summing up of things. They can look in other ways for enrichments. And that's what makes them so powerful here. And so, basically, I'm gonna hide this chat. There we go. And yeah, we decided to try, there's two methods we're using. I'm gonna tell you about this one first, and then I'll tell you about the ALS one. We'll spend most time there. I'll just go through this one kind of quickly. This showed that this kind of approach could work. We took on a disease, abdominal aortic aneurysm, which is semi-common, not as common as ALS. But it's, for people over 65, it's the 10th leading cause of death. 90% of the time it's discovered when people's aorta ruptures and they die, which of course is a terrible way to discover you're at risk for a disease, is when you die. So we obviously need early detection mechanisms. And that's one reason I wanted to put this in here. It reminds me a lot of ALS. You want to know who's at risk for it. And in this case, there's actually things you can do much better for controlling that. Like ALS, it's highly heritable. It's very, very strong genetic component, but none of the, very few genes and no definitive genes were identified, and surprisingly, again, in spite of its importance. So we did genome sequencing, meaning we determined the DNA sequence of 268 people who had this AAA, it's called abdominal aortic aneurysm, and then healthy people who didn't have that, 133. And what you do, I won't get into the details of this, but we could, go into the genome sequence. We called these these mutations that basically inactivate genes. We called them, we all have these, by the way, we all have a certain number of pathogenic mutations. And then we can mix in a little bit of electronic health record information, things like cholesterol and stuff like that. And we said in an agnostic fashion, here's the power of machine learning and this AI. You can say, give me all the genes that are enriched, linear or not. Give me all the ones that are enriched in the cases relative to controls. And it worked really, really well. 60 genes came out. I won't show you this, but we're quite confident these are right. With a very high, statistical significance, 10 to the minus 13. That's big. And the genes are involved in aneurysm and blood circulation. It wound up telling us quite a bit about AA. It was quite useful. And we could even go on and set up a predictive model for this. Now, we're not there for ALS, you'll see in a minute, but But the genome sequence alone can predict at some reasonable level, this 0.7 is actually pretty decent. And you mix in a little electronic health record, you can even do, this is a pretty decent prediction model for predicting who's at high risk for this disease. It's better than a lot of coronary artery disease predictions. So this we thought was putting us in the right track. Okay, so this was good. So we could look at these rare variants and see which ones are contributing. So you say, well, all right, well, this is great for the rare variants, but again, most diseases are thought to be due to these common changes. I should keep on the variants. Those are these changes in your DNA. How can we use that and get at this? And this is like just fantastic timing because we've been talking about this for a bit, and Jonathan wrote out to us, who's on this call, Jonathan not Cooper, or sorry, Cooper not, sorry, Jonathan. And he knows 10 times more about ALS than me, but we know how to analyze genomes. So we think it was a pretty good match. And so he says, look, we have this cohort that we want to analyze. And I don't remember if you called us up before this machine learning AA paper afterwards. But anyway, we got together and came up with a new plan for doing this. Now, this gets a little science geeky, but I think I can still tell you the important parts here. So what we did, what Jonathan mostly did, I guess with some help from us, was you can make motor neurons. So, right, your ALS is thought to involve your motor neurons, as I said at the beginning. And there's ways of doing it from these stem cells. They're called induced pluripotent stem cells. And this is why science is so advanced these days. You could never have done this 15 years ago. It's all new. And Answer ALS is actually making a lot of these lines you may know from patients. So anyway, we're basically we made these, and it's known that these mutations, for these complex diseases, they often sit in the control regions of genes. And I won't get into this, but there's a way to map these out with new methods, some of which our lab invented. And so we actually did that. We mapped out all the control regions in these motor neurons. And then we basically said, all right, we have these control regions. And we have from what are called these association studies where people tried to map the genetics, and they came up with 7 loci before. So when we entered this study, there were 7 known genes for ALS mapped by these they're called genome-wide association studies. And they had scores all the way across the genome. And so we could try this new method. Basically, the method involved, you take all the regulatory regions and you take all these sort of genetic risk scores. And this is the power of genome or machine learning. You mix them together. And again, you look for these enrichments. You look all across every one of these basically control regions. You look for an enrichment of a GWAS, of a disease association signal. And it turns out it works. We had, it's very computational intensive, it costs a fortune. But it was worth it because this is how many cases and how many controls we had from the cohort. I'll tell you about the end. And this let us map these signals. Again, with standard methods, there are only 7 genes that were found. And when we did it with this new machine learning method, that's the power of these new, again, computational methods. It worked really, really well. We went from 7 genes to 690 genes, right? So 100 times more genes were found, which is pretty incredible. And we did find a lot of genes. Some of you, those of you who follow this sort of thing, that these are famous genes in ALS. We did find a lot of them. And we could show if we take anything that anybody ever guessed was involved in ALS, that's this curated list, we could show there was a strong enrichment for those genes in our set. And other methods as well. And we compared our method with other methods out there, and it's just way, way better. So the bottom line is this new method was just a great way, shows the power of computers again for finding these things. And I mentioned before that the standard methods for looking at these genetic heritability had found 7 genes, and it turns out that accounts for 6% of the heritability. So barely, and we know remember the disease is highly heritable, but only 6% have been identified, which tells you we're missing most of the information. And then when we got to our 690, we could show using genetic methods, we actually captured a third, 33%, which turns out is pretty good for a complicated disease. Actually, it's really, really good. So, yeah, so we feel like we've gotten a nice picture, if you will, into a lot of the genetic architecture. And let's see how we're doing time-wise. Oh, we're okay. So basically, we could go on, and I'll just go through this quickly and validate that the genes were what they should be. They're really important for ALS. And I won't show you all the methods, but we can show that these genes tend to be higher expressed. We call them Refmap genes, the ones we discovered. They tend to be higher expressed in motor neurons. We could show that. And it turns out in ALS patients, they're actually more crippled. They're not expressed as well. So they're probably not functioning as well, and that leads to the defect. Okay, we could also say, what kinds of biochemical pathways do these genes work in? Are they involved? I mentioned Alzheimer's had immune genes. Well, it turns out these genes were highly enriched for things involved in the distal function of these motor neurons. So we not only figured out motor neurons, people obviously knew already, but we could show it's a very distal portion. That's where these genes operate in. We're the most critical ones for ALS function. So that's actually a novel biological insight. It says it's the very ends, these synapses, if you will, that are having trouble here, and the genes are expressed lower. So this is why they're presumably not functioning as well. And then we had this list of genes, and we went through the top ones. We did validation by means I won't get into, looking at other cohorts and looking for these rare variants. We used some of the same methods I mentioned earlier. And we zoomed in, and we had a couple of that we liked. This one we liked because it was really at the top of the gene. This gene, CANK1, was very, very high on our list. And it showed up in several different kinds of these genetic analyses we did. And so you may or may not know, so you can make these stem cells, and what I mentioned before, you can differentiate them into motor neurons, meaning you can turn them in, got to watch out for that jargon. You can turn them into motor neurons. And we meaning Jonathan's group, went in and actually made their new methods. You may have heard of CRISPR. That's one of the methods. You can go in there and actually make an ALS mutation inside this CANK1 gene that is exactly the same mutation that shows up in patients. And then it turns out it has all these problems that are very typical of ALS patients. And I won't show you all this because, again, trying to keep this a little bit high level. But this is really, really powerful. Again, you could not have done this, not easily, even probably 5-6 years ago. This ability to make these changes by CRISPR, the ability to make stem cells, this is all new, relatively speaking. It's pretty incredible when you think about it. Even turning these things into motor neurons, these are all big advances. And so we could study, if you will, in a Petri dish, what's going on with these motor neurons that had this CANK1 change. And as I say, they had all these problems that you would expect from ALS patients. And this just shows one of them, that they don't get as excited. You actually can stimulate and get neurons excited in a test tube. And neural ones with this particular stimulant will show high excitability, but these mutant ones don't. And that's, again, they're not working properly is what it comes down to. And this is kind of a key experiment for us. So a lot of people believe that these aggregates, and this is the most famous one, TDP43. I wish I could see your face. I'd ask you to raise your hand how many heard of that. But I'm assuming for those of you who are kind of geeky, you probably know that this is kind of a marker for the disease. It's actually, and a lot of people think it's the cause of ALS. But what's powerful about what we did in our CANK1 mutants, where we made this mutation, TDP is no longer properly localized like it would be in the control neurons. And so why is that a big deal? Well, what it means is that it means the mutations in the motor neurons, the motor neuron distal parts, are upstream. Forgot to say, but CANK1 operates in the distal region. They lie upstream of TDP43. So that means this is downstream. It doesn't mean it can contribute still to the disease, but it's not the most upstream cause of the disease. These distal motor neuron defects are actually upstream of TD. So it's getting to an earlier cause, if you will, of the disease. I don't know if that makes sense to people, but that turns out to be a big deal because when you're trying to find markers, you often want to get up as early in the pathway as you can. So just to wrap up this part, and then I'll say a few more comments in a minute. What we did was we discovered, from 7 to 690 genes, 100-fold more genes involved in ALS. And it's a 5-fold enrichment in this heritability, the genetic underpinnings of the disease. And we did it using a brand new machine learning method for analyzing these genetic association studies, they're called. And I think I put a slide in a minute. We actually, that's showing this is a great method in other ways. Our data basically shows that the initiation, genetics is always thought to be at the beginning, lies in the distal axon, so the distant end of your nerve cells, if you will, and that this dysfunction in these neurons lies upstream of this marker that everyone's using as a marker for ALS. And we also went on to establish that this is, in fact, a risk gene for ALS. And Jonathan should comment, I think he said that it's in a couple % of ALS patients now, even as a strong one of these single gene things. So you might ask, what's next? Well, we want to study more genes than KINK1, so we want to analyze them just the way we analyze KINK1. And one of my major goals is just this. We want to build predictive models. We're not there yet, but I know we can get there. And love Sly to comment on that, but if he's on. But we should be able to build predictive models. So just from a genome sequence, maybe with a little bit of other data, some of these other markers you mentioned at the outset, we should be able to predict who's going to get ALS pretty early on, again, potentially at birth. And that would be very, very powerful, we think, ultimately, as we get better treatments and things for treating people early and who are at risk. Ideally, we treat people prophylactically as we get cases. I won't get into this, but I'll tell you, we've actually just used this model as a really complicated slide, but we use the same principle I just told you about to actually look at severe COVID. So many of you, obviously older folks, when they get COVID, their immune system's dampened, and so they can wind up in a hospital or die. But there's a lot of young folks who get COVID and wind up in the hospital. You might say, what's going on there? Why do these folks get that? And most people think it's due to the genetics. And we've actually extended everything I told you. This general method let us pick out all the regions. We found over 1,000 risk genes for COVID severity, and we can map it onto the cell types. And here we even found a whopping 77% of the heritability for genetic risk for severe COVID. And we could actually identify the cell types, as I say. Turns out T cells and natural killer cells. That's a big deal in this field. So that is another. So we think this method will be very, very powerful. So I could wrap up now, or I could make a few comments on wearable devices. This is an area I'd like to see us move into, but I haven't seen used effectively in ALS. What do you think? Should I take a couple of minutes, Lisa, or you go through this? Or
you know what? I think it's important, the whole wearables and sensors. I would like to see a quick overview of that.
All right. Yeah, this is about 3 minutes. Okay, good.
Yeah, I'd love that.
Okay.
Thank you.
All right. So our lab is big time on wearables. Aside from genetics and genomics and other big data, this has turned out to be a really big field. We got into this when people are using this just as fitness trackers, and we realized they're pretty powerful physiological measurements. So you may know that 20% of the US population, 50 to 70 million people, are wearing a smartwatch. And they're powerful because they make hundreds of thousands of measurements on you every day. It's 365 days a year, as long as you keep them charged. I myself wear 4 of these. If you look up, you can see I'm wearing 4 smartwatches. I wear all kinds of other devices, usually use them, and they measure all kinds of things. Heart rate and heart rate variability, very, very powerful. I'd be awfully surprised if these don't have a role in ALS, and then measure other things, respiration, other sorts of things as well. And again, some of these are quite accurate. Some are accurate, but the changes are easy to mention. And I'll tell you, we're using these for all kinds of things. I'm involved with a company that's using it to measure epilepsy and other things, onset, and it can predict epilepsy before the attack. Our latest big push in our lab is actually we can detect COVID using a smartwatch prior to symptom onset in 80% of cases. And here's one example here. This is an individual who basically, here's their diagnosis day, here's their symptom day. it turns out the resting heart rate jumped up 9.5 days prior to its symptom onset. It's a very obvious signal. We've written a real-time detection algorithm for finding this. And it works actually pretty well. If you want, sign up for a study, love to have you. And so it works again on 80% of people, a median of 4 days prior to symptom onset, 7 days prior to diagnosis. So we think this is a big deal. Obviously, I think it's very relevant to this population too, as an early disease detector. And then we went on to write a real-time alerting system where you actually, you have to still ping on it. Soon we'll send you the alerts automatically. Right now, you have to ping on the app and it'll tell you whether you have a red day or a green day. Red day means there's a high stress day, which could be COVID or other things. And this is just an early example. Here's someone who actually had got their symptoms here, was diagnosed here, but got their red day 3 days ahead of time. And I can tell you recently, I just got COVID and I made the mistake of listening to my COVID antigen test. My smartwatch gave me a red alert and my antigen test told me I actually was negative. And I got on a plane, which is a big mistake. I wound up stuck in a hotel room because I, in fact, got tested positive the next day and couldn't leave my hotel room. So we think this real-time alerting system is actually more powerful than even a COVID test for early detection. Now, it's not as specific because other things can cause red alerts. And so it even picks up asymptomatic cases. Here's an example here. It works for Fitbit, Apple Watch. So feel free to join our study. So anyway, I can't imagine this not being powerful for ALS. You may mention your interface with smartphones and social media and things like that as a measure. And I think that's fine, but I actually predict that, especially for neurological disease, neurodevelopmental and neurodegenerative diseases, I can't imagine that smartwatches wouldn't be very, very powerful as a digital marker for health in the case of ALS. And so we'd love to get involved in starting some studies. I know I talked with some Answer ALS folks about this earlier. They were a little pessimistic back then. I don't know if they still are, but we could see. Anyway, that's what we're doing with wearables. We can make other predictions too about people's glucose levels or red blood cell count, stuff like that. Again, using machine learning. These are going to be very, very powerful devices. So I just want to give credit. This is just a small fraction, but it does have the major leaders here. Sai Zhang is this talented instructor who in the lab who really drove this work. And of course, Jonathan, if you don't know him, you should. He knows everything ALS, at least from my world. And it's just been a wonderful collaboration. And so we've been able to, I think, pull this off thanks to the wonderful talents of these folks. Phil Sal is another contributor. And Jonathan has a whole team working on this, many of you may know. So I think I saved plenty of time for questions. And so happy to take some if you have any.
Absolutely. Well, first, Dr. Snyder, thank you so much for presenting. I know a bunch of us will be rewatching and learning again because there's lots of information. I do want to mention that this is exactly what we're doing when it comes to wearables. Our research at Everything ALS specifically is focused on digital biomarkers. So you couldn't have spoken more to a more proud and happy to hear it team. But we will start with questions. We also definitely had Dr. Kubernox come and talk to us a couple times. So very well acquainted and happy to see him again. So myself, as well as one of our student ambassadors, Brian, will be kind of moderating the questions from the chat. The first question is, researchers attribute 2% of ALS to SOD1 genes and 4% to C9 gene mutations. Do you by chance know what percentage of the population has these mutations in the general population?
I don't off the top of my head, but it would be pretty easy to follow. There's now a database of it's called Nomad that has actually the frequency of all these gene mutations that maybe Jonathan knows for those particular ones. I personally don't, but we can look them up to see what fraction have mutations. If they're what are called highly penetrant, really disease-causing mutations at a very high frequency, they won't be much in the general population if they're truly damaging mutations. But sometimes, these mutations, they don't, like even BRCA1 will only cause disease in 50% of the cases. And so some diseases, I don't know enough about SOD, what the penetrance of those phenotypes are. Jonathan, do you know off the top of your head?
SOD1, you're dead right, Mike. So SOD1 mutations is a very, very rare in the general population. So because it is pretty much 100% penetrant. C9 is a little bit different. So penetrance is variable, and trying to understand why some people get it and some people don't. And some families look more penetrant than others is sort of focus of active research. In our population, it's about in the UK, it's about 1 in every 2,000 of the general population have a C9 mutation, whereas in ALS patients, it's about 1 in 10. So it gives you a bit of a comparison.
Wonderful, thank you.
The next question is, can CRISPR be used to fix defective genes?
Yeah, good question. I mean, for single gene mutations, people would like to get there, but the complication, of course, is these are motor neurons. So you would have to get them into the motor neurons and do just that. I think what people are hoping to do, again, others can comment, probably others on this call, but you can actually, there's 2 ways to possibly go. One is the direct gene transfer. That's expected to be low efficiency, even if you could get targeted. But I think people are very interested in ideas that would actually, where you correct the stem cells, these iPS cells, and try and add back stem cells. And so that's one of the nice things about being in California. There is a big stem cell initiative to try this, especially on neurodegenerative diseases. And so I think people are interested in that. I'll be honest, I've seen a lot more work in the Parkinson's space than I have in the ALS for this particular kind of gene correction. But I'm not as sure what's going on for ALS and the gene correction side. Again, if someone else would like to comment, Jonathan, I don't know if you know.
I think there's a couple of efforts using ASOs, antisense oligonucleotides, to sort of knock down expression of particular genes. But yeah, gene correction, like you say, Mike, yeah.
One of the nice things, though, I might add that I do know is going on. I know people are doing small molecule, meaning compound chemical screens. And this is where these IPS models are very, very valuable. We don't know whether the same molecule will work on, let's say, SOD versus CANK1 versus other things. So the more models we have like this, the more screens we can do, and the more we can test these different compounds to see if they will be effective broadly, or just be specific for certain individuals. So there's a lot of chemical screening. I forgot about that. So underlying capturing the genetics and building these model systems will be powerful in other ways. Maybe not gene correction, but hopefully chemical fixing.
We'll take whatever we can get at this point.
Yep.
You spoke in your talk about the protein aggregates, and a question came up about how do you determine what's the chicken and what's the egg? Meaning, how do we find out if maybe one of these aggregates is the cause or maybe is not? I know you spoke a little bit about TDP43 maybe being a later part of it, but is there a way to ever determine that?
Yeah, I mean, again, these model systems, the iPS cells are good for this. The reason we would say it's later is that if you can make a mutation upstream and affect the TDP43, it means there are upstream events. Now, that doesn't mean the TDP43, it could be a downstream effect, and it can still lead to some of the problems, okay? So that is possible, but it means it's not the most upstream event. And it starts helping us map out the pathway, which can ultimately be powerful, especially when you start thinking about cures. You always want to be, you want to do diagnosis as early as possible, and you want to do cures as downstream as possible often. Well, you can do it either way, but depends exactly on the nature of the mutation. This is why we got to understand this complicated disease, because knowing it will help you understand where to intervene in very specific cases. Great question, though.
Next question is, so one of the participants asked, they said, I have mutations on the MATR3 and the VAPB. What are these genes responsible for? And what kind of problems and deficiencies may they cause if there are mutations on these specific genes?
Well, I would have to look them up, and I'd probably want to understand exactly your mutations to know how to give you advice. This gets to be very tricky business. I'm not sure who gave you the diagnosis here. Some mutations are not necessarily disease causing. So you really have to look these over carefully. And there's a whole process for this. We have some very expert genome analyses, and we could take this offline and talk about it if you want. But again, maybe Jonathan or others know more about these particular genes. I don't.
Yeah, MATRN3 is sort of thought to be involved in RNA processing, and I think VAP-B is sort of vesicle transport. So both pathways associated with ALS. But I think Mike's right. Like, we have to take it's not just each gene, it's each mutation bit by bit, and be sure, because there's lots of precedents for people pulling genetic variants that aren't necessarily pathogenic. Just, you've got to be really careful, basically.
Yeah, one way to add to that is you can take your exact mutations and look again, see how often they're in the general population. If they're very common, they may not be disease causing. Now, they could if they're in combination with other things. And that's why it gets a little more involved. But if you like, I guess we could, I'd bring in my genetic counselor and we'd have a go over it with you if you like. Okay.
Our next question is, are there any longitudinal studies that you know about looking at anti-inflammatory diets and its ability to affect the genetics related to ALS, specifically C9?
So the answer is we haven't done any nutrition studies. Again, maybe Jonathan or Sai know this around ALS, but we do a lot of nutrition studies in general to see what is associated with inflammation and what's not, believe it or not. We just had a fairly big paper out looking at the effects of fiber. And we're doing a whole lot of studies around the general effects of certain nutritional things like fiber, because you may know a lot of the US population doesn't have even nearly enough fiber. Actually, the world population, we're way down. You're supposed to have women 25 grams per day, men 35, and the average US person has something like 12 to 15. Not even close. And so we're doing a ton in the space, but I don't know much about ALS. We're very interested in this. I have type 2 diabetes, so I'm very focused on diabetes and metabolic control through nutrition. And thanks to Jonathan, I'm getting pulled into ALS. I actually am a big believer food is medicine. So I would love to get at questions like yours. And to be honest, we have ways of directly measuring in real time inflammation. So I think this could be very, very powerful.
Absolutely.
So next question is, so once the gene is correctly isolated, how would it be repaired to fix the mutations?
Yeah, well, like I say, there are methods now. If you've ever heard of CRISPR, it's a way of actually editing the genome. And so these days, the way you would do it, you would actually take stem cells, either someone else's, but ideally your own, that you could derive. You can go in and correct the mutation. And as I say, in an ideal world, if everything were perfect, the people are doing this in blood, for example, to try to cure blood disease, including sickle cell, where you can actually just add the gene back directly into blood, or you can actually make the gene correction in a petri dish in the cells and then add those cells back. And for neurodegenerative diseases, the latter is more likely, meaning make the gene mutation in stem cells and then try and add those stem cells back is what most people are thinking. Now, I don't know enough about motor neurons to know if that's the right approach. I know for Parkinson's and things, it's definitely the way people are going.
Interesting. One of your slides said that the cost for full genome, or that to run genome sequencing was about $1,000. Do you have an estimation of what it costs to run everything?
Yeah, believe it or not, that's an overestimate now. Now it's we pay $500 for research genome. It's about to get a lot smaller this year. So get ready. Genome sequencing will get very, very inexpensive. Now, the whole cost of what we doing, the profiling we doing, that actually costs millions, believe it or not. That's a research study. But we, I'm in Silicon Valley, and when we want to scale these things, you form a company. So I have a company called QBio that actually does a medical version of this profiling, and they charge $3,500. They do whole body MRI and then a deep medical testing. And actually, believe it or not, they've caught a lot of stuff already, including early pancreatic cancer in folks by these, again, these profiling while you're healthy, all pre-symptomatically, they find these things. So that's what it's costing now. I wouldn't say it's cheap, but it is what it is. It's 35 and up. I think it'll get cheaper in the future as we get better and the technologies advance.
Next question is, so similarly, you spoke about polygenic risk scores. Is this something that people can do through their primary care provider?
Definitely not. There are, believe it or not, 23andMe used to give polygenic risk scores to talk about your probability of getting diseases. And most people didn't really quite know how to understand. The FDA shut them down because they didn't do it quite right. It's coming back now through 23andMe, but there are a number of academic institutions, including Stanford, that are starting to incorporate these polygenic risk scores into predictions, but it's all at the research stage right now. And so they're going to see how well it works. As I say, the research studies make it pretty clear that if you're in that top 2 to 3%, they actually are pretty decent, but otherwise it's not so useful.
When it comes to this new model, that the computer model for predicting genes, is there in the works to share that data with other ALS researchers, and do other ALS researchers share their data with you?
Yeah, so we've been fortunate. I think everybody's been fantastic about sharing data. We've reached out, and most groups have definitely come through. We shared our data early on. We put that list up of 690 genes when we first got it, and it took us over a year to publish. I'm not quite sure, maybe it was 14 months, maybe it was longer. And so we actually put it up there, and then researchers were using it right away and found it valuable, including a number of people that have reached out both to me and to Jonathan about this. So we've always shared our data. I'm actually a believer if we can get people to open consent, meaning have their data totally open and not put behind firewalls. You'd be amazed at how little that's done. You'd actually, we could do a ton more if we could get access to primary data. So it's very frustrating. We're actually pretty good about both open consenting our patients as well as sharing it, but not all groups do. And most groups, they blame it on the patients. It's rarely the patients, usually the researchers are trying to basically hold the data. It drives me nuts. It's one of my biggest complaints in science.
Yes, that is one of the things with all, I will say, our research, and I'm glad to hear that you are sharing yours too, is that for all of our Everything ALS research, it has that open data sharing model. And it is part of the game that we play, and won't start a new study unless that is all people and parties involved are agree to that. So I'm glad we are on the same page with that.
Yeah, I don't know if you're open consenting all your patients. It would be great if you do, meaning can they put it all the way up without having to put it, say, in DB gap or behind a firewall, because it's just a pain to do that. But
yes, and according to Adu, she just texted me real fast. She's like, yes, we do that too. So
all right, great.
It's good to hear.
So next question is, what are the current next steps for your research?
Well, we definitely want to analyze more of the genes. I mentioned KINK1, we knock out and see. But for me, I really want to set up predictive models. I'm quite confident we can do it. I also think we can, you may have heard me say we look at rare variants that cause disease, and we look at these common variants that cause disease. And I have new models that combine both, so we can get as predictive as possible. I'm very confident that people will get their genome sequenced before they're born in the future. And I want to be able to predict as early as possible what people are at risk for. And I think if you add in a few other markers as people go along in their biology, we will get very, very good at predicting disease well in advance. And so that's one of my big goals, use genome sequencing and other big data, again, to predict disease risk, catch disease at its earliest time. And I'm quite confident we can get there. And so the next step is really build better predictive models. I showed you the one for AAA, and Sai assures me we'll get there on the ALS. So I ask him about every month, how we doing?
For people on this call that may be either early diagnosed or soon to be in the process of potential diagnosis, what genetic tests do you recommend? Sometimes you might be asked to only pick certain panels. So Is a full genetic sequencing truly the best?
Well, not, yeah, I mean, if you're just trying to make a diagnosis on in the current clinical environment, you would use a gene panel. But I do think if you want to contribute to research for the better good of all, try and get in Answer ALS, or what, I'll give you a little plug here. So, get your genome sequenced that way and contribute to the broader good, because I still think there are many genes to be discovered. And so I think that would be fantastic. I don't know if Answer ALS has a mechanism of returning information back to folks. I think we should get to that point. I'm happy to talk to who's ever leading the genomics effort there and see what we could do, because especially once we get predictive scores, I think that would be very, very powerful. And then people like you could be the first ones we try it out on to see how well we're doing on our predictions. See if you come out high or low. If you have an ALS and you come out low on our scoring, it means we've got to go back to the drawing board some more. So so you would be a great test for us. Again, ideally, I want to have prospective studies where we make predictions and see what happens, see how well we do. But right now, I'm happy to do retrospective cases too. And the other thing we want to do is we want to subtype the disease. We want to predict severity. We want to predict response to treatments. There are obviously drugs coming along, I think all of you know. And I don't want to see the mistake where drugs work on some people, but not others, and they get set aside because they didn't work on enough people. But if you can substratify the population they work on, I'm a believer complex diseases usually aren't one disease. They really are multiple diseases. That we can see which drugs work on which people. Again, we can probably save drugs that might save lives. So I'd like to see us go that direction. So we've got plenty to do ahead of us.
Absolutely.
Yeah, you said about Answer ALS, Michael. No, they do not. We cannot give it back to them because the consent was set up in such a way that there was no return of results.
Yeah, I think going forward, it would be better
exactly.
to set that up in a way that if, you can't put an excessive burden on the researchers because you can spend all your time returning data. But I think where it's possible, if we were to spot SOD mutations or these highly penetrant mutations, it would be nice to have a pipeline that you could return it back, assuming check the box.
Yeah, we're in the process of designing the genomics sequencing one, so we'll work with you on it. That's
Great.
One of the motive is to return the results.
Terrific.
Thank you.
Yeah, we have a whole system for doing that. That involves genetic counselors, and happy to involve physicians too, if that's what we usually do. But
perfect, would be awesome.
Our next question talks a little bit about the wearables that you discussed. Do you think that the action potentials generated in neurons would eventually be able to be picked up by smartwatches and could be related for or used as a measurement tool for ALS and maybe motor neuron?
Yeah, I'm 100% sure of it. Why do I say that? Because you can pick up people's gait with a smartwatch. You can pick up all kinds of information. I don't know much about heart rate variability, but you may know that's showing a signature in almost every disease, including cancer, what have you. It would be hard for me to imagine that there's not a signal of that coming from a smartwatch. And then the other thing I didn't talk about, but we do a ton in this space, but not much in ALS. That's why I didn't. But I'd like to get this is an area I'd like to push. We're actually getting pretty good at this stuff, if I'm allowed to say that. But I'm pretty confident there's something called galvanic stress response. There's no clinical correlate for this. What it does is it measures conductance on your skin. So I'd be awfully surprised if that doesn't have a bit of a signal in this particular case too. So as I say, we can get a signal on diabetes on that. And that's simply because diabetics have drier skin, and you can pick that up pretty easily with this kind of measurement. And at the end of the day, it turns out it's multiple kinds of measurements. I may not have mentioned, but respiration is one thing you can pick up. So imagine people shifting in their breathing patterns. I'm pretty sure, in fact, 100% confident I've seen already in patients we've looked at. You can pick that up. So, and then you just integrate over all the signals, you build machine learning models. And I'd be awfully surprised if we can't get quantitative measurements for ALS and possibly even substratify again the disease. I don't know, but that would be a goal.
Wonderful.
Next question is, once the gene mutation is corrected, what can then be done to restore the damaged neurons?
Yeah, well, as I say, one hope would be that maybe there's a way to transplant these in a way that would restore function. I know that's harder probably with motor neurons and other, neural developmental diseases like Parkinson's, but that would be one goal. People are working on specific targeting vectors to try and introduce genes into specific cell types. I suppose that's possible too. But I don't think we're even close to that for ALS. So unfortunately.
We know now looks like 690 potential mutations for ALS. According to your experience and expertise, what do you think may be the main cause of these mutations?
Oh, the main cause of mutations? Some of them are natural variation that are out there that are probably modest in effect. Others, yeah, they probably are more damaging mutations that can completely inactivate gene function. I don't know. It's kind of interesting. To be honest, preparing for the advantage of sucking a person like me into the field is I know nothing. And so I always look at things with a fresh face. And when I see something like in preparing this talk, again, 1 in 300, why is that? If you think about other cases where there's these highly frequent mutations like cystic fibrosis, they're thought to be protective, okay? And so is there a protective function to ALS? Obviously not the way we see it, but are there mild forms of ALS that are actually helping you in certain circumstances? I don't know. I'm throwing that out. That was a question I asked, believe it or not, while watching this, because what do I know about ALS? But every other disease, sickle cell, right, has a protective function. Yeah, and cystic fibrosis supposed to help people with TB was one possible model. And there's other things like this as well. Almost every frequent disease out there, there's some reason why it's there that helps people in certain kinds of situations, like even CCR5, which is, it's a receptor for the AIDS thing. You think, well, if you could get rid of it, you don't get AIDS, it must not have a real function, but it probably helps in certain kinds of situations. So I'll challenge this group here to think about what are the protective circumstances for having, some risk for ALS. Obviously, the debilitating form is not going to be good, but I could envision that maybe some mild forms protect you in certain situations.
What do you think about, motor neuron death is like kind of part of aging process, but would ALS be super aging where, you're having, the death of motor neuron at the rate where you're super aging? What do you think about that concept?
Well, I like it in the sense that, most oxidative stress leads to neural damage and stuff. So, and anything that increases oxidative stress and other sorts of things that I think are related to this do lead to aging. And you're probably thinking a lot of the sarcopenia where you lose muscle mass and motility and things like that. I don't know if you lose motor neurons though in sarcopenia. I just haven't looked into that. So it's an interesting concept. And certainly oxidative stress, I think is a bad thing. You guys must know though whether antioxidants improve ALS symptoms. Is that true or not?
Yeah, it's true.
Yeah. So I could envision that, especially with the SOD link. Yeah, and that will then relate back to nutrition. So I don't know if everybody's here is taking NAC or NAD sorts of things that those are some of your best antioxidants out there.
Absolutely. I think we have, go ahead, Brian.
Go for it, Sarah. You sure? Okay. So the last question is, do any of the wearables accurately measure oxygen during sleep? And if so, can you recommend any that do?
Well, I wouldn't say smartwatches do a very good job for accuracy. Again, they're good for deltas. They'll pick up changes. There's new smart rings out there, and I don't know how good they are, but for sleep, you could probably just wear, again, I just had it here. Oh, here it is. Yeah, you can go buy one of these things. These are these pulse oxes you put on your finger, and you can just probably sleep with one of these things if you want an accurate one. It probably won't fall off, or it might fall off after a few hours. But yeah, assuming you can sleep with one of these, they're not that invasive. So you can just buy one of these online. I have a Massimo, and I have no. By the way, I'm conflicted in everything I told you, probably, so you can ignore it if you want. But I have no stock or any thing in Massimo. So those are the most accurate ones, I think.
Wonderful. Well, Dr. Snyder, we want to thank you very much for coming today. This has been very informative, and I hope you've gotten a chance to go through the chat, but people were very excited about your presentation and very thankful for all your explanations. I will laugh to tell you that it was for a genius genetics guy, it was very easy to follow. So that's always a good pat on the back for yourself, because sometimes we've all been in genetics class where that's not the case. So we appreciate that. But we want to say thank you one more time, and look forward to all your future research, and hopefully you'll come back and present again, and we get to see next big steps.
Okay, yeah, no, I am getting, thanks to Jonathan, he's got me sucked in here. I'm very hooked, actually. So I hope to do a lot more studies. I did put my email in the chat. I saw someone ask for that, so feel free to reach out. I'll probably just pass the question on the side, Jonathan, anyway, but I'll give it a shot. So
Well,
thanks
good.
again for having me.
Absolutely.
Thank you, Mike.